Good afternoon, ladies and gentlemen. The topic of the lecture is teaching about infection. And it's very important topic, and there are many medical terms you should know. And teaching about infection. What is it at first? What is it, infection? The term infectious disease applies when an interaction with a microbe causes damage to the host, and the associated damage of altered physiology results in clinical signs and symptoms of diseases. So if some person has clinical symptoms and some results in human body, we are talking about infectious diseases. And every infectious disease is caused by certain pathogen. A pathogen is usually defined as any microbe that has the capacity to cause diseases. Not all pathogens have an equal probability of causing diseases in the same host population. There are different pathogens. Principal pathogens regularly cause diseases in susceptible individuals with apparently intact defense system. And of course, they are not in, principal pathogens do not belong to normal microbial flora. Some species are commonly part of normal microbial flora of humans and animals, but also can frequently cause diseases, and they are named conditional pathogenic microbes. They cause the defects of the passive carried to the internal environment of an organism. Opportunistic pathogens are those that really cause diseases in immunocompetent people, but can cause serious infection in immunocompromised patients. These opportunists are frequent members of the body's normal flora. The origin of the term opportunistic refers to the ability of the organism to take the opportunity offered by reduced host defensive to cause diseases. If normal microbes, those are able to exist like normal microbial flora, have opportunity to multiply, they will multiply, and finally infectious disease will be developed. And such infectious diseases are named opportunistic infections. Some microbial pathogens are obligate intracellular parasites. For example, viruses, some bacteria such as chlamydia and rickettsia, because they can multiply only within the host cell. All stages of life cycle of microbes are connected to an organism of the host. Many microbes are facultative intracellular parasites because they can multiply within cells, outside cells, or on different media. It doesn't matter for them where are they, out of human cell or maybe inside. A part of the life cycle, facultative parasites spend as free living organisms. Nevertheless, the parasitic phase has conducting value. Facultative parasites, many intestinal bacteria, Neisseria, tuberculobacteria, Brucella, causative agent of brucellosis, are casual parasites, free living organisms. They entering in the organism of the host is casual, absolutely an essential phase of life cycle. They don't need really the human's body or animal's body, and they enter human's body occasionally. Casual parasites are fungi that exist in the soil. They cause mycosis or bacteria of Legionella genus that exist in the water, and they are able to cause pneumonia and fever, sometimes very severe, named legionellosis. Pathogenicity, very important term. Pathogenicity defines the ability of a microbial species to produce the certain infectious diseases. Like, for example, species name is Vibrio cholera. And exactly, bacteria those belong to species Vibrio cholera are able to cause cholera. Bacteria those belong to species Bordetella pertussis are able to cause whooping cough. They are not whooping cough, and patient has very long-lasting coughing. In case of cholera, patient has vomiting and diarrhea. So every species of microbes is able to cause specific diseases, and this it is ability to cause specific disease, ability of species. And virulence is applied to the same property in a strain of microbes. Because of strain, those belong to one species sometimes differ by this ability to cause infectious diseases. So, and the ability of certain strain, those belong to some species, is named virulence. The term reflects a degree of pathogenicity, various strains of the concrete pathogenic microbe. Some strain maybe have are more virulent compared with another. Some of them maybe, some strain maybe are virulent strain. They are not able to cause disease in spite of all strains belong to one species, but they are differ by this ability. And virulence provides a quantitative measure of pathogenicity of the likelihood of causing diseases. It is possible to estimate the ability of every strain to cause infectious diseases. As a unit of measurements of virulence, the lethal dose DL from the in Latin, dosis letalis, was accepted. What does it mean, dosis letalis? Dosis letalis is the least amount of pathogenic microorganisms of the toxin capable to cause death of the certain quantity of laboratory animals. Of course, this ability to cause infectious diseases is estimated by using of laboratory animals. So we should use many groups of laboratory animals, and we should prepare different doses, like amount of microbes per 1 cubic centimeter, for example. And then this dose should be injected, different doses should be injected at different groups of animals, and we will see what is the least amount of microbes that is able to cause death of the certain quantity of laboratory animals. And there are different doses. Doses certa letalis in Latin, in abbreviation is DCL, DCL, quantity of microbes or the toxins causing death of 100% of experimentally infected or intoxicated laboratory animals. If in some group completely the 100% of animals did die after using of this dose will be doses certa letalis. LD50 quantity of the microbes or toxins causing death of 50% of experimentally infected laboratory animals. Apply also doses LD70, LD90, and so on. Very often, the virulence of different strains is compared by using of LD50. And if for some strain LD50 takes 100 microbes in 1 cubic centimeter, and for another strain it takes just 10 microbes in 1 cubic centimeter, what does it mean? The second one is the most virulent strain, because just 10 microbes is enough to cause the death of 50%. So it is possible to compare doses, and we should realize which strain is more virulent. DLM, doses letalis minima, quantity of microbes or the toxins causing the death of 95% of laboratory animals and group. Doses letalis minima. For doses certa letalis, it should be 100% of laboratory animals, the death of 100%. In case of DLM, it is necessary to see the death of 95% of laboratory animals. It is possible to compare virulence, and why it is important, and really it's important in practice, because of, then you will study vaccines. Vaccines are used in medicine for specific prophylaxis of infectious diseases. And some vaccines consist of live pathogenic microbes. And it is possible, of course, they are used, but we should decrease the virulence. The virulence should be decreased by 13 factors. These microbes, these vaccines consist of special so-named vaccine strains of pathogenic microbes. Louis Pasteur was the first scientist who did create vaccine for specific prevention of anthrax, at first for animals and then for human beings. And he did prove that it is possible to decrease the virulence. He did cultivate this bacteria, Bacillus anthracis species bacteria, in unfavorable conditions at very high temperature, and some bacteria did die finally. But some of them did survive, but they lost virulence. They're still alive, but they were not able to cause anthrax. It was he was genius really, because before him, all scientists believed that it was not possible to change the properties of microbes, and he did prove that it is possible to decrease the virulence. And he did developed a specific vaccine for specific prophylaxis of anthrax. And after him, another scientist, Calmet and Guerin, he did develop vaccine for specific prevention of tuberculosis. It was at the beginning of last century. At that time, many people did die of tuberculosis because of that time doctors had no any specific prevention and no good preparations for treatment of tuberculosis. So they did create the vaccine from the Mycobacterium bovis. And again, this is kind of live vaccine. In this vaccine, bacteria is still alive, but they lost the virulence. So, and we should check the virulence. These vaccines are produced, they're commercial, they're industrial. And these vaccine strains, and after that many vaccines were produced from bacteria, from viruses, like measles vaccine, rubella vaccine, mumps vaccine. They are live vaccines. And and some bacterial vaccines consist of live microbes. And so these vaccine strains are cultivated, and before packing we should check the virulence, because the thing is that due to mutation, sometimes maybe these microbes will reverse to original shape. And we should always check the virulence, and we have to be sure that they have very mild virulence, that exactly they still vaccine strains. Okay, so it's important by using of laboratory animals. Very often LD50 is used for detection of virulence. Then some term is used, infectious dose, ID. Infectious dose is another, do not mix please. It's not lethal dose. In infectious dose is a minimal quantity of the pathogenic microbes capable to cause development of the diseases at the certain number of experimentally infected laboratory animals. And for every causative agent, no, for most of causative agents, infectious dose is estimated, and it is different. So realize that if 1 or 2 bacterial cells enter human body, even tuberculosis bacteria, for example, or Vibrio cholera, they're not able to cause infectious diseases. Called in a human's body, there are many defensive factors, specific factors, non-specific factors, and due to these factors, this small amount of microbes will be eliminated, will be deleted from human body, and they have no chance to multiply and to cause diseases. So, and for many causative agents, the infectious dose is estimated. It is different. The lowest infectious dose is for hepatitis B virus. It's enough to get just a tiny, just a 100 part of the drop of the blood, and it is enough to be infected by hepatitis B virus. So infectious dose for hepatitis B virus very small. For some microbes, maybe it estimates like 100 microbes, maybe 1,000. So difference. But anyway, big microbes should enter human body in infectious dose. And if there are a lot of microbes enter human body, so they are able to multiply quickly, to spread in human body, and finally they will cause clinical symptoms of disease, okay, due to some pathogenic factors. Then factors of pathogenicity, major factors of pathogenicity of microbes. We told a lot about different properties of microbes, of bacteria. Now we are talking about bacteria, and we did study morphology, staining properties. You did study cultural properties, biochemical properties, and now you will study factors of pathogenicity, virulence factors, okay? So, and number one, you should learn by heart all of them. And number one, adhesion. When pathogenic microbes enter human body, the first task for them is to attach to the human's being cells, because otherwise they will be removed again due to some many factors and adhesion. Then invasiveness. Invasiveness is ability to enter in inner part of the body of host. They should go inside, okay? Then stability to different microbicidal factors of host. They should protect themselves from phagocytosis. They should protect themselves from immunoglobulins action. So, and they have many different ability to protect themselves. They are able to create a capsule. They are able to destroy immunoglobulins. They produce proteases, enzymes. Those are able to split immunoglobulins and so on. They are able to produce enzymes for protection. So protection factors. Then ability to produce some toxins. And ability to produce some enzymes of aggression, tissue degrading enzymes, and then ability for a long persistence. Now we will talk the most frequent portals of entry of pathogenic microbes into the body are the sites where mucous membranes meet with the skin: respiratory, upper and lower airways, gastrointestinal, primarily mouth, genital and urinary tracts. It is easier for microbes to enter into the human body through mucous membranes. Skin is a mechanical barrier. They are not able to go through the skin if it is intact without any lesions. But it is easier to go through the mucous membrane, to attach to the mucous membrane. So abnormal areas of mucous membranes and skin, cuts, burns, and other injuries are also frequent sites of entry. If skin is damaged, of course, it is for microbes, it is easier to go inside. Normal skin and mucous membranes provide the primary defense against infection. And to cause diseases, pathogens must overcome these barriers. Adhesion factors. The number one, they should attach to the surface, adhesion. Once bacteria enter the body of the host, they must adhere to the cells of a tissue surface. If they did not adhere, they would be swept away by mucus and other fluids that bathe the tissue surface. And adherence, which is the only one step in the infectious process, is followed by development of microcolonies and subsequent steps in the pathogenesis of infection. Many bacteria have pili and fimbria, we did study, hair-like appendages, that extend from the bacterial cell surface and help mediate adherence of the bacteria to the host surface. So fimbria, many bacteria have fimbria for adhesion. Then, invasion of host cells and tissues. How are they able to invade? For many disease-causing bacteria, invasion of the host epithelium is central to the infection process. Some bacteria, for example, Salmonella species, invade tissues through the junctions between epithelial cells. Other bacteria, for example, like Neisseria gonorrhoeae, Chlamydia trachomatis, invade specific types of the host epithelial cells and may subsequently enter the tissue. Some bacteria, for example, Shigella species, multiply within host cells, whereas other bacteria do not. So invasion is the term commonly used to describe the entry of bacteria into the host cells. Then toxins, very important part, toxins and toxins produced by bacteria are generally classified into two groups. So memorize, bacteria are able to produce exotoxins and endotoxins. And this is characteristic of exotoxins you should learn by heart. Exotoxins, what are they? In biochemical nature, they are heat-label proteins. Heat-label in general, but there are some exceptions, like botulotoxin is heat-stable and enterotoxin produced by staphylococci, unfortunately, is heat-stable protein, but there are exceptions. Mainly, these exotoxins are heat-label proteins, so it is possible to destroy them by using high temperature. Action often enzymic. They act like enzymes, and they have specificity, specific tissue affinities. So they are able mainly to destroy, to act to destroy the certain cells. Like for example, some exotoxins are able to destroy red blood cells. They are named hemolysins. Some of them are able to act on cells on digestive tract. They are named enterotoxins. Some of them are able to destroy this upper layer of skin, exfoliative toxins, and so on. So, and they are some of them are able to destroy many cells, membrane toxins, they are named. Some of them are able to destroy tissues, they are named histotoxins. So they are named because they have specific tissue affinity. Active in very minute doses, they are very, very toxic, highly antigenic. They are recognized by immune system, and specific antibodies named antitoxins are produced. And of course, they are detected in case of toxinemic infections and readily separable from the cultures by physical means such as filtration. It's possible to it's possible to separate them, and it is possible to produce from them. It is possible to produce specific preparation for vaccination named toxoids. So exotoxins, not all of them, but for in case of 7 infectious diseases, exotoxins are used for industrial production of toxoids. Toxoid in English, anatoxin in Latin. So many gram-positive and gram-negative bacteria produce such toxins of considerable medical importance. And 7 infectious diseases are named toxinemic infections because of circulation of exotoxin in blood leads to death. And they are staphylococcal infections, diphtheria, tetanus, botulism, gas gangrene, whooping cough, and cholera. And such circulation of exotoxin in blood is named toxinemia. So such infectious diseases are named toxinemic. But there are another causative agents. Those are able to cause, to produce exotoxins, but not very often it leads to death. These 7 diseases are very dangerous because of toxinemia, exotoxins action. And many exotoxins consist of A and B subunits, and the B subunit generally mediates adherence of the toxin complex to a host cell, and ACE entrance of the exotoxin into the host cell. And the A subunit provides the toxic activity. And some of these toxins have had major roles in world history, like diphtheria, outbreak of diphtheria, and so on. Okay, cholera is pandemic infection, and all these diseases are very common toxinemic infections. Circulation of exotoxin in blood is known as toxinemia, and toxinemic infections, the most dangerous are tetanus, botulism, gas gangrene, diphtheria, whooping cough, cholera, and staphylococcal infections. But in case of another diseases, like in case of typhoid fever, disease caused by Escherichia, exotoxins are produced, but not very severe diseases. These diseases are the most dangerous, okay? So vaccines have been developed for prophylaxis of the exotoxin-mediated diseases, and these vaccines called toxoids are made from exotoxins which are modified so that they are not longer toxic. And then we will study how it is possible to okay, the exotoxin should be isolated, bacteria should be cultivated in liquid medium, and they will produce exotoxins, proteins, and we will separate them by using of filtration, membrane filters, and we will get proteins, and we will get toxic protein. And the task is to keep antigenic structure, but to decrease the virulence, to remove toxic properties. And two factors are used. Formaldehyde is used like chemical factor, and this mixture should be placed at thermostat. And another factor, physical, high temperature should be used, 37 degrees by Celsius during 38 days. We should cultivate this mixture. After that, toxic centers of these exotoxins are blocked by formaldehyde, but protein is not destroyed. And finally, we will get the same protein without toxic properties. And it is named toxoid, and it is used for vaccination against these 7 diseases: tetanus, toxoid, botulism, FU toxoid 3, gas gangrene 2 toxoids are produced. There are different species: diphtheria toxoid, whooping cough toxoid, cholera toxoid, and staphylococcal infection. Toxoids are used for specific prevention. Pharmaceutical faculty student, you should know preparations better than other students. Okay, so there are properties of exotoxins. You should learn by heart. And then there are properties of endotoxins. What are they? The heat-stable lipopolysaccharides, LPS, and of gram-negative bacteria are derived from the cell walls and are often liberated when the bacteria are destroyed. We did study structure of gram-positive and gram-negative bacteria, and you know that exactly gram-negative bacteria have got lipopolysaccharides in the upper layer. They have proteins, lipids, and polysaccharides. So exactly, lipopolysaccharides are toxic for human beings. Abbreviation is LPS, I told you. And but they are not produced specially. For production of exotoxins, by the way, it is necessarily the tox plus gene is necessarily. And exotoxin is not life important. Exotoxins are not life important for bacteria. And if bacterial cell has got tox plus gene, this bacterial cell is able, transmitted by bacteriophages, by the way, transduction and so on, you know, and a bacterial cell has got tox plus gene, information is encoded about toxic protein production. But if a bacterial cell has no this tox plus gene, exotoxin is not produced, and everything is normal for bacterial cell. Bacteria don't need exotoxins. So memorize, and because of in case of diphtheria, some bacterial cells have got tox plus gene. They are able to produce exotoxin. They are named toxigenic strains in case of diphtheria, and some of them are not have no tox plus gene, so they are not able to produce exotoxin, they are named non-toxigenic strains, and they are not able to cause diphtheria. And a person may be infected by non-toxigenic strain of Corynebacterium diphtheria, and never clinical symptoms of diphtheria will be developed during all the life. So it depends on the presence of tox plus gene. Endotoxins are LPS. They belong to the cell wall of gram-negative bacteria. So all gram-negative bacteria, without an exception, have got LPS. So they have endotoxin. And the action of exotoxin, so they are differ. Endotoxins are differ. Endotoxins are differ from exotoxins. Number one, by biochemical nature, they are lipopolysaccharides, and they're heat stable. They are not antigenically active. We are not going to detect antibodies for LPS. Maybe they are produced, not so many, and not important. Okay, and not possible to prepare toxoids from them, and they have no organs affinity. Lipopolysaccharide action are just in common. Doesn't matter. Is person infected by gram-negative cocci, gram-negative rats, and anyway, the clinical symptoms are common. The pathophysiological effects of LPS circulation in the bloodstream can be observed clinically or experimentally on laboratories like fever, leukopenia, and hypoglycemia, hypotension, and shock, resulting in impaired perfusion of essential organs like brain, heart, kidney, and death from massive organ dysfunction. If a lot of microbes, gram-negative bacteria, multiply in human body, like for example, meningococci, a gram-negative cocci, I hope you remember, and the meningococci have just LPS. They are not able to produce any exotoxins, but if they multiply a lot of in human body, even in spinal cord, they are able to cause the death. It was meningitis, leads to death of patients. Very difficult to save the life of this patient. So just somewhat this action, okay? So, and all memorize all gram-negative bacteria have got LPS, so they have endotoxin. And what else? Tuberculobacteria. Tuberculobacteria are not gram-negative bacteria. If we will stain them by gram staining, we will see violet colors. So it is detected that they are gram-positive bacteria, but they have lipids in the cell wall. Those are toxic for human beings, and intoxication is developed again due to lipids action of the cell wall of tuberculobacteria. Spiral bacteria such as Leptospira borrelia, they do not produce exotoxins, and they are not stained by gram staining, but by in the cell wall, again, they have lipids. So by structure of the cell wall, they are close to gram-negative, and again, intoxication is developed due to lipids action. Rickets and chlamydia, again, they are close to gram-negative bacteria. Of course, Giemsa staining is reduced for them, but anyway, they have toxic substances in the cell wall, so they will cause the same pathophysiological effects in patients. Then, enzymes. Bacteria are able to produce tissue degrading enzymes, and like Clostridium perfringens produces the proteolytic enzyme collagenase, which degrades collagen and promotes spread of infection in tissue. Staphylococcus aureus bacteria produces coagulase, which works in conjunction with blood factors to coagulate plasma. Plasma is liquid part of blood, but due to presence of coagulase, the plasma will be solid, becomes solid, and every cell of Staphylococcus aureus is carved by this coagulated plasma. So this bacteria will get false macrocapsule that is consist of coagulated plasma, and they are disappeared. They are not recognized by immune cells. They are not available for antibodies. So because of coagulase production, this is pathogenicity enzymes, of course. Coagulase contributes to the formation of fibrin walls around staphylococcal lesions, which help them persist in tissues. Coagulase also causes deposition of fibrin on the surfaces of individual staphylococci, which may help protect them from phagocytosis or from destruction within phagocytic cells. So they are not recognized staphylococci. Really, they have no capsule, but they are able to create capsule from the plasma inside the human body. And many bacteria produce cytolysins. They dissolve red blood cells, hemolysins, or kill tissue cells, or leukocidins, hemolysins, so cytotoxins, and immunoglobulins A proteases. Immunoglobulins are produced by plasma cells, and their specific antibodies, their synonym is antibodies. Immunoglobulins are antibodies, and immunoglobulins A are the secretory antibodies on mucosal surfaces. And some bacteria that cause diseases produce enzymes named immunoglobulins A proteases that split IgA and inactivate its antibody activity. So again, it's a very production of immunoglobulins A protease allows pathogens to inactivate the primary antibody found on mucosal surface and thereby eliminate protection of the host by the antibody. So immunoglobulins A are on the surface of mucosa. They are first antibodies that meet bacteria and are able to neutralize them. But if bacteria are able to produce proteases, they will split immunoglobulins, so and they are not neutralized. Okay, so and now specific features of infectious disease are specificity. Every disease is caused by certain causative agent. Infectivity means the patient is dangerous. Microbes are spread from one patient to another people. And cyclicity. Every infectious diseases go through incubation period, then prodromal period, then the period of development of clinical signs, and then stage of increase of clinical size, stage of a peak of illness, stage of a decrease of clinical signs, and then probably via crisis or slow disappearance, and maybe a reconvalescence. Reconvalescence is a completely recovery when clinical symptoms are disappeared and the microbes are eliminated from patient body. Reconvalescence, real recovery, but maybe chronic form, maybe carriers form, and maybe the death. It's okay. So, and forms of infectious disease, and so I have no time to explain everything. So we will discuss on laboratory classes. Have you any questions? Have you any questions? So now
No,
you
doctor.
should answer the test on educational port. I will check attendance of the lecture due to answers for the test lecture, okay? Now on education portal should answer the test.
Okay.
Leaders, could you inform all students from your group if someone dismissed the lecture? Anyway, all students should answer the test, okay? Sure. If you have no questions, so, Just 32 students. So everyone should answer the test on educational portal. Otherwise, students
Okay.
should do the work of missed lecture.